Well, thank you to the organizers for inviting me. This is always very exciting to give a talk, especially in front of an audience that's halfway around the world. So my name is Ho Leung Ng. I'm at Kansas State University in the US. I'm a professor in the Department of Biochemistry and Biophysics. I welcome you to contact me by email if you have any questions later about my research or my talk. So where am I located? So we are in the state of Kansas. This is in a agricultural area in the middle of the United States. Our university is most well known as a agricultural university, although my research is primarily not in agriculture. We are the site for the U.S. National Bio-Agro Defense Facility. So this is one of the focuses of this research facility is to study zoonotic diseases that can be spread that are relevant to agriculture. This would include diseases like COVID-19. However, this facility is still being built, and unfortunately, we could really use it right now, but we are not quite ready. My research background. So thank you for the very nice introduction. Most of my research background is actually not related to COVID-19. I do work in structure-based drug design, primarily in cancer, immunology, and malaria. I started work on COVID-19 in earlier this year when I was contacted by Chinese collaborators who sought some assistance. And I felt that this was a place where I hopefully could make a difference.
Hello,
Uh,
Dr. Who.
yes.
Sorry for the interruption. We
Oh,
are
my
not able
slides
to
are not
see the
changing.
slide change. Yeah, yeah,
I see. Okay, I did not realize that. Okay, so let me.
yeah. Now it's fine. Yeah, it's changing.
Okay, sorry. Yes. Uh, okay. So we're talking about my research background. So my background is primarily in structure-based drug design. This is both from a computational and experimental perspective, using in silico tools, developing our own methods, as well as wet lab biochemistry, biophysics, crystallography. And in the last about 2 years, we've been uh, working very hard in developing and applying machine learning to drug design. So one of the uh, initiatives I'm most excited about is OSC 19, Open Source COVID-19. So this was a group that I started earlier this year. We now have over 20 members across the world. This is a group dedicated to nonprofit open source drug discovery, meaning that we will not have patents, and we will share research data openly to promote drug discovery, and especially to promote affordable drug discovery that can be accessible to a global population. Our group is inclusive. We're open to all scientists that have a background that is relevant to this kind of research. If you're interested, please contact me. Our capabilities run from computational drug design all the way to chemical synthesis, biochemical assays, crystallography, and we are one of the few consortiums that have access to actual COVID virus, SARS-CoV-2 viral testing in a BSL-3 level facility. So the current targets in our AI drug discovery program are the MPRO protease and NSP16 RNA methyltransferase. So you've already been introduced to these two targets by other lecturers. So I will not go too much into detail on them. We will focus primarily on MPRO protease today because that is considered the most important and heavily studied SARS-CoV-2 drug target. So the main strategies that we are pursuing, one is virtual screening of drug bank for purposes of repurposing. So repurposing is very important to us because this is probably the fastest way to get a drug to humans to actually help patients. As we all know, unfortunately, every day thousands upon thousands of people are getting sick and people are dying. So every day counts. So what we're doing is we're screening Drug Bank, a database of characterized drugs, drug candidates, etc. Doing not only many people are doing something similar to that. So the way that our approach differs from other groups is that we're not just screening Drug Bank, but we're screening the adjacent chemical space of Drug Bank, meaning that if we find promising hits from Drug Bank, we'll also screen molecules that are that have high similarity to those hits, because those molecules, although they may not be characterized experimentally and are not drugs themselves, are more likely to share the desirable chemical properties of drugs. The other thing that we are doing that is different is that we are doing virtual screening for covalent inhibitors. So most people have focused on screening of non-covalent inhibitors. That is rather easy, but the methods for screening covalent inhibitors is not so well developed and generally requires quite a lot of human expertise. So it can't be done in an automated manner. So we have done that, and we have completed the virtual screen. One thing that is important, following on the lecture, our previous speaker, is that our results are being validated experimentally. So we are in the process of validating our in silico results using enzyme assays and crystallography, and the best results from those will go on to be tested against live virus. But this is something that you probably by now have learned quite a bit about. So what I want to do is talk about something that's a little different, which is that using artificial intelligence methods to generate novel inhibitors. So this is something that we're doing. This is different from repurposing. Unlike repurposing, this is probably not the most efficient way of pushing towards real drugs. However, this is important because this is a way of developing the latest technologies for future drug design. So let me give you a very brief introduction to how AI is used for at least the chemistry of drug design. I don't want to make this too technical. So nowadays, the method that has excited the most people is called deep learning. So deep learning can be considered a subset of machine learning, which is a subset of AI. Machine learning uses primarily tools that are based on statistics, and deep learning uses a very specific kind of tool, which are deep neural networks. A deep neural network is a neural network that contains multiple layers for calculation. So what is a neural network? So again, this can get quite technical. So I just want to give you a very brief introduction so you can understand what this is about. They are difficult. They look difficult, but actually the way they work is rather simple. You have these circles, and we call them nodes, and each circle corresponds to a function that is applied, that is calculated from inputs. So an input here is shown as x1, x2, and x sub n. And each of these inputs can be weighted by and so here's W1, W2, Wn, etc. Your node calculates some kind of function. And then the output from this function, which here is y sub j, is then fed to the next level of uh, of of nodes. And so you do this, and each vertical alignment we call a layer. So you see these multiple layers, and eventually you calculate them to the output layer, and then that gives you your final result. And so neural networks have been used for computing for decades, but it is only very relatively recently that we have developed deep learning neural networks and have learned how to use them in a way that is useful. And the power of deep learning neural networks comes from their ability to act as something called a universal approximator. Basically, a deep neural network can represent any function. So a function here in the broad sense means that from a set of inputs, one can calculate some kind of output. So basically, for example, you could think of all the inputs that you would feed into a neural network that would predict whether a given molecule would be a useful drug or not. And then you can train your network, meaning you parameterize it based on experimental or simulated data. And then once that network is trained, you can apply it to new data, and hopefully it will have some kind of predictive power. So we talked a bit about that. So there are two main AI applications to drug discovery. So one is predicting drug activity from chemical structure. So this turns out to be a very hard problem that people have been working on for decades. So it is a problem that is rather simply stated that here's a chemical structure. Can you tell me whether it would be effective as an inhibitor against M-pro protease, for example? And it turns out that this is quite hard, especially if you want to get quantitative results, such as to actually predict the inhibition constant or the free energy of binding. The second application, which I'm going to focus on today, is something that's a little bit, that's actually even harder, but is something that is closer to the heart of the leading edge of AI-driven drug design, where not only are we making predictions, but we're asking this computer to use those predictions to actually design new molecules that hopefully will have better properties that make it more drug-like. So I'll talk about that a bit more, as that is something that is not as familiar to a general audience. Okay, but let's talk a little bit about the first application, because to be able to do AI-driven design effectively, we need to be able to make useful predictions about drug activity. So how do we calculate protein-drug interactions? So we start with extracting the chemical features. So this word features is actually a technical concept in machine learning. And all it means is that we just want to pull out data, any kind of data that might be useful input that can be fed into a neural network. These features would include relatively simple chemical properties, such as molecular weight or calculated hydrophobicity, electrostatic charges, etc. And one can use, the nice thing about machine learning is that you can calculate and use as many chemical features as you want and feed them into the neural network. And part of what the network does is that it tries to calculate the weights and the relative significance of these different features. So you have your neural network architecture, you feed it the features, and now you train your network on different data sets of correct and incorrect uh, docking orientations, meaning that where you have a given molecule, and then you have to take into account both the conformation of the molecule, the conformation of the target, and how well the drug molecule fits the drug target. We identify those significant features that are correlated with the correct docking, as well as the inhibition constant or the free energies of binding. And then we use the neural network to help us create functions that are predictive. So we call these functions scoring functions. So there are different kinds of scoring functions that are broadly used. And to introduce them to you, so we have some that are called force field based. Another term for this is physics-based models, meaning that they try to calculate a binding affinity based on traditional physical chemistry types of parameters, such as Coulomb electrostatics and van der Waals interactions, etc. Now, this turns out to be quite difficult. And in most, this turns out to be quite difficult. Another way to do this is instead of using physics-based parameterization, we use empirical parameterization. We use a model that's somewhat physics-based, but we parameterize certain constants against data sets from experimental binding affinities. And in general, this is currently the tends to be the most effective way, especially combining it with the force field-based models. Another way to uh, determine scoring functions is to use knowledge-based potentials. What is a knowledge-based potential is that instead of, for example, using experimental binding affinities, we can use actual observed distributions of physical chemical parameters, such as interatomic distances. We can get those from crystal structures. And this is nice because this provides an orthogonal method of of scoring compared to the first two. And so what generally works the best is to combine the best features of these three. And you can do that very nicely by using neural networks. And so as a relatively simple but very effective example of how this works is that we were able to do this in the D3R, the Drug Design Data Resource Competition that was held 2 years ago for predicting drug activity. And by using a relatively simple neural network architecture where we combine structure-based features, and these would be features that are based on three-dimensional interatomic distances and properties with ligand-based features. So these would include the simple parameters such as ligand molecular weight and hydrophobicity. We're able to calculate experimental binding affinities quite accurately with an R correlation coefficient of 0.67 for base, which is a important drug target for Alzheimer's disease. So what we want to do is apply these kinds of methods to the current challenge of COVID-19 drug discovery. So now we will move to the second application of AI to drug discovery, which is using generative AI modeling. So this ties in to the big question, the bigger topic of inverse design. So inverse design means that we're first thinking about functional space. There are certain chemical and physical properties that are of interest to us. For example, solubility, toxicity, drug activity. Now, how can we map from functional space to chemical space? So we have the desired properties, but to actually get a drug, we need to be able to get a real molecule that can be synthesized and put into a vial and given to patients. So there are different ways one can do this. One can do this traditionally through doing enormous amounts of experiments. We can apply this data. We can do in silico modeling, as we discussed earlier. And another way of doing this is by using simulations and by using what we call generative modeling. So generative AI or generative machine learning. Using the machine learning algorithms to learn how chemical space and functional space map together. What properties are correlated with each other, and using that to directly move into molecules. So this is a problem that is general to chemistry and goes beyond just drug design. So here's one way to think about it. You might be more familiar with some of the work in machine learning on images. So big companies like Google and Facebook have a lot of interest in analyzing faces. And so here are a set of human faces. And what's interesting about these faces is that these faces do not correspond to real people. These are faces that are computer generated. So the computer algorithm has looked at an enormous data set of human faces. And the algorithm is able to learn what a human face looks like without a human saying, well, a human face usually has two eyes and a nose down the middle, et cetera, et cetera. The machine learning algorithm can learn this. And this is actually quite old. So you can, if you look carefully, you can find artifacts in these that make them that you can give away that these are not real human faces. The most current algorithms can come up with human faces that, at least to me, look completely real, which is quite scary. Now, we take this same approach, and instead of thinking about faces, we ask the computer to think about molecules. We give a computer a set of training molecules, and we can say, hey, these molecules, first of all, the computer does not know what is chemistry. It does not know the rules of chemistry. So the computer has to learn that, okay, we are just starting with the balls and sticks, and they have to connect in certain ways. And they connect in certain ways, and that gives you a molecule. And this molecule, we feed the computer, we label these molecules with certain properties. And we tell the computer, from these molecules, we can calculate certain properties. So computer, learn chemistry, and then also learn what are properties associated with molecules that would make for good drugs. This is very hard, as you might imagine, but there are lots of advantages for this kind of strategy. One, first of all, you must realize that chemical space is enormous. What do I mean by chemical space? I mean the number of different molecules one could make that occupies a certain molecular weight range. So, for example, let's say the number of molecules that one could make that are below 600 in molecular weight, which characterizes most small molecule drugs. The number of molecules that could make are almost infinite. So how can we, if they're infinite, so that means we can't screen them directly, even in silico. So how can we explore the chemical space in a way that helps us find drugs? So based on our knowledge and experience with medicinal chemistry, we could say, well, the space is infinite, but we know from our research that we think this region of space is the most promising for making drugs, or this region of space, and especially for a drug targeting, let's say, M-pro protease. Now, of course, if it was that easy, then we'd be making new drugs all the time. So it turns out it's not that easy at all. We can make some educated guesses so that it's not random. But as anyone who has experience in drug design research knows, that there is an enormous amount of trial and error involved. And this trial and error takes a long time and is extremely expensive. So what this kind of AI, generative AI can do is that it can help us explore chemical space. It can help us move away from a human expert's a priori guess at what might be the most promising space to less biased regions of chemical space. Now, just like our previous speaker said, this is not a tool that will move us automatically to a drug. What we're trying to do is we're trying to improve chemical diversity, especially in a way that is not biased and often unexpected from the perspective of a human expert. So we think that's an advantage that a computer can have. The computer is not smarter than us, but perhaps it can avoid some common human biases. So now we're moving to the current state of the art. So one of the things that our collaborators have found is that in addition to the actual machine learning itself, that the data representation is extremely important. So here's a molecule, and the traditional way that chemoinformatics scientists have represented these molecules is by using something called a SMILES string. And here's an example of a SMILES string. The SMILES string, unfortunately, has a lot of drawbacks. And our collaborators developed a method similar to, which extended the concepts of a SMILES string that they called SELFIES. What a great word, huh? And what the advantage of SELFIES does is that the SELFIES contain the complete topological connectedness of the molecule, and it also includes some rules about what kind of atoms can and cannot be connected. And what that does is that instead of having the neural network have learn what is the rules of chemistry, the grammar, the data representation, the SELFIES themselves contain the simple rules, and that helps the neural network it frees the neural network to work on the harder and more interesting task of helping us find interesting drug-like molecules. So the actual architecture, which we won't focus too much on, that uses a genetic algorithm. And what this does is that it introduces new molecules and it mutates them, modifies them, and crosses over their good features. It uses a neural network, which is trained on a data set and continues to be trained on molecules that are generated to try to discriminate between molecules that are good or bad. It also uses properties that we tell the algorithm that we want. For example, we don't want molecules that are too small or too large. We don't want molecules that are too polar or too hydrophobic, etc. And so this is great because this can include anything that can be quantified. So the properties and the output from the discriminator are fed together into a fitness function, and then that gets fed back into the generator to create the next generation of molecules for evaluation. And what we can see here is that over multiple generations, this hybrid architecture is able to improve improve by lowering the fitness function. So it's which means the error in the fitness function, as well to improve, for example, the likelihood that they would rank high for some kind of desired chemical property. So back to MPro. So this is all work that has been done just very recently within the past week. And what we're starting with is almost nothing. We're starting with a methane molecule that is put into that is evaluated at the active site of the MPro protease. After 10 generations, the algorithm is able to expand the methane to various molecules. So there are actually hundreds of molecules in each generation. So these are just some randomly chosen examples. And after even more generations, what we see here is that the molecules continue to grow in size and complexity. So from our limited testing so far, these are examples for MPro of molecules that scored quite high in this by this algorithm, meaning that these molecules fit a certain desired properties that we told it to fit, as well as they tend to dock relatively well into the MPro binding site. And if you, for those of you who are chemists, you'll find that these molecules, most of them look okay, but they do have some parts that look quite strange. So, and that is something that we're still working on. Also, many of the molecules that are generated are not what we call synthetically accessible. So these are molecules that might exist on paper, but are not easily synthesized by a chemist. So that's not of practical use. But we can still use these because we can feed these as ideas to a human expert, and the human expert can say, I never thought about using something like this, and but of course, this is not feasible. So the human expert can modify this into something that is usable. Another way to do this that's more simple is there are various different kinds of simplifications that we can apply. So we take this molecule and we look for an analog for an existing molecule in PubChem, which is a large database of chemical structures. And we found that this analog of that previous molecule actually scores quite well and docks relatively nicely into the MPro protease binding site. Okay, so as I said before, this is something that we're working on this very minute, and there's still a lot of room for improvement. But this is something that is close to the state of the art. So, of course, like I said earlier, we want to make molecules that are more drug lead-like, meaning that they show more properties that are characteristic of drugs. We want to improve synthetic accessibility because this is not just a computational exercise. We want to have molecules that can be put into a vial. Instead of, as a proof of concept, we started from the most difficult case. We started with a methane molecule. Let's make it easier. What if we start with known drugs or known active compounds or fragments and ask the algorithm to see if we can use those and as a seed and build better molecules? And more generally, can this kind of algorithm be used as a test of the druggability of a given target site? Because this is done in an automated, relatively unbiased manner. If the computer algorithms cannot come up with a molecule that can give good binding, at least in silico binding to a site, can we conclude that this is not a promising site for testing, and vice versa? So I want to acknowledge the work, the people who have worked on this. So the computational lab at our university, these are the graduate students and postdocs in my lab who have worked on this project over the years. A lot of the experimental work in OSC 19 is being led by Troy Messick's group at the Star Institute in Philadelphia in the US. Our collaborator in machine learning is Professor Alan Asproguzic at the University of Toronto, and these are his students and postdocs who have led the generative machine learning work. Our research is currently sponsored by the United States National Science Foundation and Intel Corporation. Thank you very much for your attention. I welcome your questions.